Dive in. Let's go. Number 10. The Mesopelagic Zone and the biomass nobody counted. In 2014, a team of researchers from the National Oceanography Center in Southampton published findings in the journal Deep Sea Research that forced marine biologists to quietly revise one of their most fundamental assumptions about life in the ocean. The study used acoustic data collected from research vessels crossing the Atlantic, and what it returned was a number that did not fit any existing model of ocean biology. The total biomass of fish living in the Mesopelagic Zone, the layer of water between 200 and 1,000 meters depth, had been estimated for decades at somewhere between 1 and 2 billion tons. The Southampton data suggested the real figure was closer to 10 billion tons. The scientific community had undercounted the fish in the middle of the ocean by a factor of 10. The Mesopelagic Zone begins where sunlight fails. Below 200 meters, photosynthesis is no longer possible. The water shifts from blue to black over the course of a few hundred meters, and what lives there has adapted to a world without light, without warmth, and without the food sources that define surface ecology. The fish of the Mesopelagic are small, mostly lanternfish, bristlemouths, hatchetfish, species that produce their own bioluminescence to hunt, to hide, and to communicate in conditions that would disorient any surface-dwelling predator completely. But here is where the undercounting becomes disturbing. The acoustic instruments used in traditional ocean surveys were calibrated for shallow water species. They were not designed to detect the specific acoustic signatures of deep mesopelagic fish at depth. The surveys had been running for decades, producing estimates, publishing figures, building entire models of ocean carbon cycling and food web structure on numbers that were wrong by an order of magnitude. 10 times wrong, and nobody knew. The implications extend further than a simple revision of fish stock estimates. The Mesopelagic Zone is the primary pathway through which carbon moves from the surface ocean into the deep. Every night, billions of mesopelagic fish migrate upward toward the surface to feed, then descend again before dawn, carrying the carbon from their meals back down into the dark. This process, called the biological pump, is one of the primary mechanisms by which the ocean absorbs atmospheric carbon dioxide. If the biomass driving that pump is 10 times larger than the models assumed, then the models of how the ocean regulates the planet's atmosphere are also wrong. By how much, nobody has yet calculated with confidence. There are more fish in the dark middle of the ocean than in every fishery humanity has ever exploited. They have been there the entire time. We simply never looked in the right direction to count them. And the ocean kept the number to itself for as long as we were willing to not ask. Number 9. The deep scattering layer and the thing that looks like a false seafloor. During World War II, Navy sonar operators began reporting a persistent anomaly that no one could explain. At depths between 300 and 500 meters, sonar returns were coming back as if from a solid surface. A false seafloor. A layer of something dense enough to reflect sound the way the actual seabed reflects it, sitting hundreds of meters above the real bottom. Operators initially assumed the equipment was malfunctioning. Then they noticed the layer moved. During the day it sat deep. At night it rose toward the surface. By dawn it descended again. It was alive. What the sonar was detecting was the largest animal migration on Earth. Every night, without exception, across every ocean on the planet, billions of mesopelagic organisms, fish, squid, crustaceans, jellyfish, rise from depth toward the surface, in coordinated vertical migrations that can span several hundred meters in a matter of hours. The layer is so dense that it returns sonar as a solid mass. The creatures inside it are so numerous that their combined bodies register as in geography. Scientists named it the deep scattering layer. The name has been in the literature since the 1940s. What it actually contains, the full species composition, the total biomass, the behavioral dynamics of the organisms inside it, remains poorly understood more than 80 years later. Direct sampling of the layer is difficult. Nets pulled through it collect some specimens but destroy others and miss the fastest swimmers entirely. Submersibles that descend through it at night describe walls of bioluminescence moving around the vessel in every direction. Animals too numerous to count, too varied to catalog, pressing against the lights from all sides before vanishing back into the dark as the submersible passes. Here is where the scale becomes genuinely difficult to process. The deep scattering layer is not a local phenomenon. It exists in the Atlantic, the Pacific, the Indian Ocean, the Southern Ocean, the Arctic. It occurs every night, everywhere, simultaneously. The total number of individual organisms participating in this migration has never been estimated with any confidence. The best current models suggest it is in the trillions. Every night, the largest coordinated movement of living creatures in the history of the planet, happening in the dark, just below where the surface light fades, while the surface world sleeps. Number 8. The Twilight Zone predators that science has not finished naming. Between 200 and 1,000 meters depth, in the layer oceanographers call the Twilight Zone, there are predators that hunt in conditions most surface species could not survive for 30 seconds. The pressure at 500 meters is 50 times atmospheric pressure at sea level. The temperature is close to freezing. The only light is bioluminescent. The only sound is the low-frequency hum of distant shipping traffic and the biological noise of the organisms moving through the dark around them. In 2020, a research expedition operating in the Eastern Pacific lowered a baited camera system into the Twilight Zone and left it running for several hours. When the footage was recovered, marine biologists cataloged more than 30 distinct species interacting with the camera over the course of a single deployment. 12 of those species were unknown to science at the time of filming. 12 new species, in a single camera deployment, in waters that research vessels cross regularly. The Twilight Zone predators have evolved solutions to the problem of hunting in permanent darkness that surface biology never produced. The dragonfish, genus Malacosteus, generates red bioluminescent light from organs beneath its eyes. Red light is invisible to nearly every other species in the deep ocean because the water column filters red wavelengths long before they reach that depth. The dragonfish hunts in a color of light that its prey cannot see. It is functionally invisible while it is searching, and the things it is searching for have no warning that it is coming. The stoplight loosejaw, a related species, has a jaw that unhinged downward to a near vertical position, allowing it to engulf prey items larger than its own head. Its stomach is black on the inside. Every surface of its digestive system is designed to prevent the bioluminescence of swallowed prey from leaking through the body wall and revealing the predator's position in the dark. But here is where the Twilight Zone stops being a collection of strange biology and becomes something that should change how you think about what the ocean is. Every species discovered in the Twilight Zone in the last 20 years was previously unknown, not rumored, not partially described, completely unknown. The expeditions that found them were not searching for new species. They were conducting routine surveys in waters that had been considered adequately cataloged. The new species were simply there, in the dark, in waters crossed by research vessels for more than a century, waiting to be looked at directly for the first time. The Twilight Zone is not a frontier. It is a neighborhood that has existed alongside human civilization for the entire history of our species. We simply never turned on a light inside it until recently. And every time we do, something new is looking back. Number 7. The giant squid and the predator that hunts it in the dark. The giant squid, Architeuthis dux, was not confirmed alive by photographic evidence until 2004. Before that date, the species was known almost entirely from dead specimens washed ashore, fragments recovered from the stomachs of sperm whales, and reports from sailors that the scientific community had spent 2 centuries dismissing as exaggeration. The first living footage of an adult giant squid in its natural habitat was captured at 900 meters depth in the Pacific by Japanese marine biologists Tsunemi Kubodera and Kyoichi Mori. The animal in the footage was hunting. It was not drifting. It was not dying. It was an active predator operating with precision in absolute darkness at pressure that would kill a human being instantly. The giant squid reaches lengths of up to 13 meters. Its eyes measure up to 30 centimeters in diameter, the largest eyes in the animal kingdom, evolved specifically to detect bioluminescent signals in the midwater column where no other light exists. It hunts fish and smaller cephalopods in the Twilight Zone and below, using tentacles lined with suckers reinforced with chitinous hooks that grip prey with a force that leaves permanent circular scars. Here is where the midwater column reveals something that the creature lists in this niche rarely discuss. The giant squid has a predator. Not a larger squid, not a shark, a sperm whale. Sperm whales dive to depths exceeding 2,000 meters specifically to hunt giant squid in the midwater column. They descend in darkness, navigate by echolocation, locate individual squid across distances of several hundred meters, and engage in fights that leave both animals scarred. Sperm whale skin is covered in circular marks from squid hooks. Sperm whale stomachs contain squid beaks that do not match Architeuthis. They match something larger. The colossal squid, Mesonicoteuthis hamiltoni, was confirmed from a complete specimen in 2007. It weighs up to 500 kilograms. Its tentacle hooks rotate on swiveling bases. Its eyes are 27 centimeters across. It lives deeper than the giant squid, primarily between 1,000 and 4,000 meters, in the lower midwater column, where sperm whales still pursue it. Researchers studying the species have repeatedly noted that the recovered specimens are not representative of the full adult size range. The largest individuals have never been observed. They exist at depths where human observation requires equipment most institutions do not possess, hunting in a darkness that has no upper limit on what it can conceal. The midwater column between the giant squid's range and the colossal squid's range contains an active predator-prey relationship between animals that weigh hundreds of kilograms, playing out in total darkness, at pressures that destroy unprotected human tissue every hour of every day. We have observed fragments of this system. We have never watched it directly, and the sperm whale beaks that keep appearing in the wrong size category suggest there is at least one more participant in that system that we have not yet confirmed. Number 6. The bioluminescent communication network nobody has decoded. In 1999, marine biologist Edith Witter descended into the midwater column off the coast of Biscayne Bay in a submersible equipped with a device she had spent years designing. The device was called the Eye in the Sea. It was a camera system built to record bioluminescent activity in the deep ocean without emitting the artificial light that normally causes deep-sea organisms to flee or freeze when a submersible approaches. What Witter recorded during that first deployment fundamentally altered scientific understanding of how life in the midwater column communicates. The footage showed continuous bioluminescent activity across every depth the camera reached. Flashes, pulses, sustained glows, coordinated patterns between multiple organisms, light produced and received in sequences that bore the structural characteristics of communication rather than simple camouflage or predator attraction. The midwater column, the space that was assumed to be dark, was producing light constantly, in patterns, across species, in what Witter and her colleagues described as a language the ocean had been speaking for hundreds of millions of years before anything with eyes evolved to read it. Bioluminescence in the midwater column serves functions that surface biology does not require. Counter-illumination, where organisms match the faint light from above by producing light from below, making them invisible to predators looking upward. Burglar alarm displays, where prey organisms flash intensely when attacked, illuminating the predator and attracting larger predators to the location. Lure systems, where predators produce species-specific light patterns to attract prey that associates those patterns with food. The diversity of bioluminescent strategies in the midwater column exceeds anything documented in any other environment on Earth. But here is where the bioluminescent record stops being cataloged biology and becomes something genuinely unsettling. Witter's subsequent deployments of the Eye in the Sea system produced footage of bioluminescent displays that do not match any cataloged species. Patterns of light produced by organisms that moved through the camera's field of view too quickly to be identified, in sequences too complex to be simple threat responses, at depths where the species inventory is incomplete enough that new entries appear with every expedition. The ocean is producing light in patterns we cannot read, from organisms we cannot name, at depths we have barely visited, in a communication system that has been running continuously since before the Cambrian. We are listening to a conversation we do not have the vocabulary to understand, and the participants have been talking across the dark water for longer than complex life has existed on the surface. Number 5. The oxygen minimum zone and what survives inside it. Between approximately 200 and 1,000 meters depth in many regions of the world's oceans, there is a layer of water where oxygen levels drop to concentrations so low that most aerobic life cannot survive. These layers are called oxygen minimum zones. They form where biological productivity at the surface is high enough that the decomposition of sinking organic material consumes oxygen faster than circulation can replenish it. The result is a band of chemically hostile water sitting in the middle of the ocean, a barrier that most species cannot cross. The oxygen minimum zones of the Eastern Pacific and the Arabian Sea are among the most extensive. In some regions, oxygen levels drop to less than 5% of surface concentrations. Fish that venture into these layers begin to suffocate within minutes. Most marine predators treat the oxygen minimum zone as an impenetrable wall. They hunt above it or below it but cannot operate inside it, which means the oxygen minimum zone is the most effective hiding place in the ocean. Species that have evolved tolerance for low oxygen conditions exist inside these layers in what amounts to a protected refuge. Their predators cannot follow them. Their competitors cannot reach them. The oxygen minimum zone functions as an evolutionary sanctuary, a place where unusual biology can develop in isolation over geological time, sheltered from the competitive pressures that shape surface and shallow water ecosystems. Here is where the oxygen minimum zone stops being an interesting oceanographic feature and starts being something that should make you reconsider what the midwater column is hiding. In 2010, researchers sampling the oxygen minimum zone of the Arabian Sea using fine mesh nets recovered organisms that had never been described in any prior survey of those waters. The specimens included crustaceans with no known relatives, gelatinous organisms of uncertain classification, and fish with physiological adaptations to low oxygen conditions so extreme that biologists studying the samples could not initially determine how the animals were extracting enough oxygen from the water to remain alive at all. The oxygen minimum zones cover tens of millions of square kilometers of ocean volume. They are difficult to sample because the equipment required to operate in low oxygen conditions is specialized and rare. Most ocean surveys pass through them without stopping. The species that live inside them are known from fragments, from net samples, from the occasional specimen that surfaces dead and unidentified. The living communities inside the oxygen minimum zones of the world's oceans have never been directly observed in anything approaching their full extent. The ocean built an invisible wall in the middle of its water column, and whatever was strange enough to survive inside that wall has had millions of years of uninterrupted darkness to become stranger still. Number 4. The marine snow and the ecosystem it feeds that nobody mapped. In 1977, marine biologist Alice Aldredge was diving in the Mesopelagic Zone off the California coast when she noticed something that had been overlooked in every prior description of the midwater column. The water was full of particles, not sediment, not plankton, aggregates, loose, fluffy, organic clumps of material drifting downward through the water column, like a continuous, slow snowfall. She collected samples, brought them to the surface, and discovered that each particle was a miniature ecosystem. Bacteria, phytoplankton fragments, fecal pellets, shed exoskeletons, mucus strands, all compressed into aggregates that hosted communities of microorganisms actively feeding, reproducing, and dying as they sank. She called it marine snow. The term has been in the literature ever since. What Aldredge had found was the primary food source for the entire midwater column. Marine snow falls continuously from the surface ocean into the dark below. Every organism that dies near the surface, every fecal pellet produced by every zooplankton, every shed exoskeleton, every fragment of dead phytoplankton, eventually disaggregates and begins its slow descent. The journey from surface to seafloor can take weeks or months depending on depth and particle size. During that descent, the marine snow passes through every layer of the midwater column, feeding the organisms at each depth as it goes. The midwater column does not produce its own food in any meaningful quantity. It receives food from above, processes it, and passes what remains to the depths below. Every organism in the Twilight Zone, every creature in the oxygen minimum zone, every predator hunting in the dark between 200 and 4,000 meters is ultimately dependent on the rain of organic material descending from the sunlit surface above. The productivity of the surface determines what the midwater column can support. But here is where marine snow reveals something about the scale of midwater life that has taken decades to begin to understand. The amount of organic carbon that reaches the seafloor after passing through the midwater column is a small fraction of what left the surface. Between 80 and 90% of the carbon in sinking marine snow is consumed before it reaches the deep seafloor, consumed by the organisms of the midwater column, organisms whose total biomass, as the Southampton study found, was underestimated by a factor of 10. The midwater column is not a passage between the surface and the bottom. It is the largest active ecosystem on Earth. It intercepts, processes, and recycles the majority of the organic material produced at the surface before any of it reaches the seafloor. The organisms doing that processing have been largely uncounted, largely unobserved, and largely unnamed. The machinery of the ocean's biological pump runs through the dark middle of the water column, and we have been standing at the surface watching the input and measuring the output without ever looking at what happens in between. Number 3. The midwater predators that migrate through depth every single day. There is a fish called the bristlemouth, genus Cyclothone. It is between 2 and 5 centimeters long, translucent, and possesses a jaw that opens to a width disproportionate to its body size. It is, by number of individuals, the most abundant vertebrate on Earth. More individual bristlemouths exist in the world's oceans than all other vertebrate species combined. The total population has been estimated in the hundreds of trillions. They live in the Mesopelagic Zone, and they have been there, in those numbers, for the entire history of vertebrate life on land. Nobody talks about bristlemouths. They are not large, they are not dramatic, they do not appear on the lists of terrifying deep-sea creatures that populate this niche. But their existence reveals something about the midwater column that the dramatic species cannot. The Mesopelagic Zone supports vertebrate populations of a scale that the surface ocean cannot approach. The dark middle of the water column is not sparse. It is dense beyond any human intuition about what the ocean contains. The vertical migrants that use the midwater column as both refuge and hunting ground include species across every taxonomic group. Lanternfish, which make up the largest component of the deep scattering layer, ascend to the surface at night to feed on zooplankton and descend before dawn to avoid surface predators. Myctophids travel several hundred meters vertically twice in every 24-hour period, their entire lives spent in this daily transit between two worlds. Certain species of squid migrate from 800 meters depth to the surface and back every single night. The energetic cost of this migration is enormous. The benefit must be greater still, or evolution would have eliminated the behavior millions of years ago. Here is where the vertical migration reveals something that should change the way the ocean is understood. The midwater column is not a place where creatures live. It is a route, a corridor, a daily commute traveled by trillions of organisms moving between the productive surface and the dark refuge below. The column is in constant vertical motion, day and night, in both directions simultaneously, with different species moving up as others move down, in a flow of biomass that has no equivalent anywhere else on Earth. The predators that exploit this migration are positioned throughout the column at depths where specific prey species are densest at specific times of day. The column is not random. It is structured, layered, organized around the daily movement of prey in ways that predators have spent millions of years learning to anticipate. And the full architecture of that system, every predator, every prey species, every timed interaction across every depth, has never been mapped. We have sampled fragments of it with nets and cameras and acoustic surveys. The complete picture does not exist. It has never existed. The column keeps moving while the instruments try to catch it. Number 2. The gelatinous organisms that dominate the midwater column and dissolve before we can study them. In 2017, the Monterey Bay Aquarium Research Institute released footage captured by its remotely operated vehicles in the Mesopelagic Zone off the California coast. The footage showed something that had been documented in fragments for decades, but never captured at this scale or clarity. The midwater column was dominated by gelatinous organisms, jellies, ctenophores, siphonophores, salps, not in the occasional scattered densities that surface observations had suggested, in curtains, in walls, in aggregations so dense that the ROV cameras struggled to see through them. The gelatinous organisms of the midwater column represent a category of life that marine biology has systematically underestimated for the entire history of ocean science. The reason is straightforward and disturbing. They dissolve. Gelatinous organisms are composed primarily of water, held together by structures so fragile that contact with a net destroys them instantly. Every sampling method developed for ocean surveys relies on nets, on trawls, on collection devices that make physical contact with specimens. Contact kills gelatinous organisms before they reach the surface. For most of ocean science's history, the organisms most abundant in the midwater column were the ones least represented in the samples. The siphonophore is the most extreme example of how badly traditional sampling has failed to capture midwater reality. Siphonophores are colonial organisms, chains of specialized individual animals called zooids, each performing a distinct function, assembled into structures that can reach lengths of 40 meters or more. Apolemia, a siphonophore filmed by Mbari in 2020 off the coast of Western Australia, measured approximately 47 meters in length, making it potentially the longest animal ever recorded. It was discovered not by a net survey, but by an ROV hovering at a respectful distance from a creature that any net would have shredded beyond recognition in the first second of contact. But here is where the gelatinous midwater community reveals the depth of what conventional survey methods have missed. The 2017 Mbari footage and subsequent deployments have documented gelatinous organisms in the Mesopelagic Zone in densities that, when scaled across the full volume of the world's midwater columns, suggest a total gelatinous biomass that exceeds every prior estimate by an order of magnitude comparable to the fish biomass revision from 2014. Two independent categories of midwater life, both undercounted by a factor of 10 or more, in the same layer of the ocean, simultaneously. The midwater column is not the sparse, poorly populated transitional zone that decades of net surveys implied. It is the most densely inhabited space on the planet, and the organisms that make it that way are the ones that disappear the moment you try to bring them to the surface to count them. The ocean has been hiding its most abundant residents behind the fragility of their own bodies. They dissolve before they can be evidence. And the column they fill has been described as nearly empty for a century because the tools used to describe it could only collect what survived the collection. Number 1. The unidentified acoustic signals coming from the midwater column right now. In 2005, researchers from the Scripps Institution of Oceanography published findings describing a previously uncharacterized acoustic signal detected repeatedly in the Mesopelagic Zone of the Pacific Ocean. The signal occurred daily. It was produced by biological sources. It was loud enough to register across significant distances on hydrophone arrays, and it did not match the acoustic profile of any cataloged species in the Mesopelagic community. The researchers named it the Deep-Sea Twilight Zone Chorus. It occurred at dusk and dawn, corresponding to the timing of vertical migration in the deep scattering layer. Billions of organisms moving simultaneously through the same depth range produced a collective acoustic signature that the hydrophones detected as a coherent sound event rather than random biological noise. The chorus was real. Its producers were not fully identified. The specific species composition generating the signal has still not been determined with confidence. The Twilight Zone Chorus is one entry in a catalog of midwater acoustic anomalies that has been accumulating since the Cold War hydrophone networks were first turned toward the ocean. The systems built to listen for Soviet submarines recorded biological signals from the midwater column that Soviet submarine analysts had no framework for interpreting and simply logged as biological noise. When those records were reviewed by marine biologists after declassification, they contained decades of acoustic data from the midwater column of the Pacific and Atlantic, data that had been collected by some of the most sensitive listening equipment ever built and then filed without biological analysis. Some of that data has since been reviewed. What the reviews found was a midwater acoustic environment far more complex than any prior biological survey had suggested. Species vocalizing at frequencies not previously associated with mesopelagic fish. Acoustic signals repeating in patterns that suggest social behavior in species not previously believed to be social. Call structures in the Mesopelagic Zone that share characteristics with the communication signals of surface cetaceans, produced by organisms that are not cetaceans and whose identity has not been confirmed. Here is where the midwater acoustic record stops being an interesting research gap and becomes the most disturbing implication of everything on this list. The hydrophones are still running. The midwater column is still producing signals. Every day, in every ocean, the acoustic environment of the space between 200 and 4,000 meters depth generates sounds that the most sensitive listening networks ever built are recording in real time. Some of those sounds are identified, many are not. The catalog of unidentified midwater acoustic signals grows every year faster than the rate at which existing signals are assigned to confirmed species. Something in the dark middle of the ocean is talking. It has been talking for as long as we have had instruments sensitive enough to hear it. The conversation does not pause when our instruments are pointed at it. It does not change when our submersibles pass through. It continues. In the dark, in the layer of the ocean we have spent the least time looking at, in a register we are only beginning to develop the vocabulary to read. We built the most sensitive ears in human history and pointed them at the largest habitat on Earth. What came back was not silence. It was a language we do not speak, coming from a population we have not counted, living in a space we have not mapped. In the middle of an ocean we have been sailing across for thousands of years without ever once looking down far enough to see what was there. The midwater column is not between the places that matter. It is the place that matters most, and it has been keeping that fact to itself for the entire history of life on this planet. If you want to see more videos like this, and make sure to subscribe.